Victoria Makerspace for a tour. He's one of the co-founders of Victoria Makerspace and one of the co-founders of the biospace in Victoria as well. And he's currently finishing up his PhD, and I will let him explain what that's about because it's very long. Thank you all for taking the time out of your Sunday afternoon, so close to Christmas, to come out and support the space and also support Derek. So without further ado, I will introduce Derek Jacoby from Victoria.
Good. Thanks a lot, Rebecca.
Thank you.
So I should mention as well that we have one of the other co-founders of Victoria Makerspace on the video conference here. Thomas Gray is up in Powell River, and a couple of the folks from the Powell River Makerspace that they're setting up there are going to join us as well. So if you hear disembodied voices coming out of the speaker, it's people talking from Powell River. Treat this all really informally. This is some slides I put together last night to guide the discussions, but I really want to treat this as much as a discussion around biohacking as I do a formal conference. So hit something I don't explain well enough or something you have questions on, please feel free to stop me and we'll talk about those. Rebecca mentioned the PhD thesis. That's been dominating my life these days. I'm in the midst of getting out of a computer science PhD at UVic, where I'm building low-cost electroencephalography devices and a protocol for diagnosing and treating attention deficit disorder using EEG-based neurofeedback to work on attentional conditions. So in a sense, a form of biohacking, and that's actually a pretty good lead into some of the stuff we're going to be talking about today, that there are these two streams of biohacking that didn't originally have too much to do with each other, but are pretty quickly converging. And so the one stream of biohacking is this definition of biohacking as cybernetics almost, where you're taking pieces of hardware, pieces of technology, and embedding them in yourself to achieve some new human capability. I've always thought that was really cool, but it's not actually something that has been at the core of my work these last few years, aside from, as I mentioned, that PhD thesis. The other train of biohacking, though, is the one that I've been much more involved in, which is about taking genetics and treating genetics as this malleable information code, changing it, putting it into organisms, and really creating new life forms. I'll talk in some depth about that and how it's actually a very appropriate activity for hackerspaces here as we get on into the talk. The fascinating thing is, though, that these two forms of biohacking have been coming together. And so a lot of what I'm going to talk about at points in the talk here are places where I've been able to use knowledge of genetics, and specifically knowledge of my own genetics, to make health and wellness, and in some cases even performance enhancements. So we really are getting to the point where cybernetic enhancement and genetic enhancement are not necessarily going to remain different for the rest of our lives. So I'm going to talk about some futures as well, particularly on the genetic side of things. There are some fascinating things going on, and over the next 20 years, we can expect to see increases in longevity, engineered babies that are going to be many times smarter than we are. All of these things that you would read about in science fiction and go, is that really real? And it's our responsibility to make sure that we tread a nice path through these futures rather than a dystopian path through these futures. And so that's one of the reasons that I think it's really important that at kind of the grassroots of technology spaces like this, we understand what these changes are so that we can help guide our leaders and guide our policymakers in weaving a path through these, where there are some very real dangers that we could fall into some of these dark dystopian science fiction horror shows. But if we don't, if we thread the needle and find our ways to kids that are many times smarter than us and capabilities many times greater than we have, and longer lives, well, there's a really rosy future available to us there as well, but it's going to take some needle threading. So that's where we'll get into the role of the hackerspace in all of this. Let me start at some of those cybernetic enhancement kind of points. This Riddick-like fellow on the right here, Gabriel Lucena, I spelled his name wrong, not Gabriel Lucena. Gabriel Lucena has been one of the folks in the news recently using a form of chlorophyll eye drops to get good night vision. This is something that originally was discovered in a cave fish species, and some analysis showed that this was kind of how they were getting their good night vision. Was picked up some by the U.S. military, a little bit of human testing by the U.S. military, and these folks decided, hey, this sounds like a good thing to try biohacking on. Let me tell you, this is where I get a bit more comfortable with my form of genetics biohacking than the physically based biohacking, because injecting a new form of chlorophyll into my eyes to give me night vision is a little bit beyond my risk tolerances. He's seeing well again today, though, and his night vision is worn off. And if anything, it was temporary effects. But that's kind of the level of personal risk and responsibility around playing with some of these things that I need to think about, though. And it really is going mainstream. On the left-hand side here, you see a bandage on Zoltan's hand after doing an RFID implant. I've got a few good friends who have done this and used them for all sorts of kind of interesting things, starting their motorcycle, opening their front door, getting their mailbox to open. But nothing that has really kind of, again, wanted me to cross that barrier to where I'd be afraid of getting my hand pinched in the grinder and breaking that little glass ampoule and going, oh shit, now I've got a broken little glass ampoule in my hand. And the benefits, the things I could build with that RFID chip have never quite seemed compelling enough to make me take that implant leap. Aside from some of that a bit of breakage risk, though, this is a very mature technology. They've been using the RFID implants for animal tracking. This is what your dog gets when you take him to the vet and want to make sure you can always identify your dog if he runs away. Is they put one of those RFID ampoules under their skin, and so they've got all the right growth factors to make it grow in solidly. And really, they've shown that over many years it doesn't cause any problems. So that's kind of on the low risk end of the physical biohacking. The story on the bottom, though, Buttermind, is one of the sad elements of biohacking. I was at a quantified self meetup in the San Francisco Bay Area about 4 years ago now. And this quantified self theme is one that I'm going to talk about over and over in different ways throughout this discussion. It's a organization. There have been four quantified self conferences now. Organization that gets people together a couple times a month down in the Bay Area, and it's spread to some other places to have presentations from members of the groups where the way they organize these presentations is by saying, what did I do? Why did I do it? And what did I learn? So it's really kind of crowdsourcing knowledge acquisition in N of one experiments. These people are experimenting on themselves and finding something valuable, trying to show that it was collected in a fairly scientific way, sharing it with people, then trying to encourage other people to join in on those experiments to turn it from N of one to N of however many join in. This is a fairly different form of knowledge acquisition than we're used to in traditional scientific circles, where anecdotal data and N of one data is really quite ignored in most of mainstream science. And you really have to have double-blind studies and large N studies before you'll really be listened to in that environment. But historically, of course, that anecdotal N of one data is how a lot of treatments have been found. Certainly in psychology, you've got this strong tradition of individual results and individual treatment plans. It's really only when you get to kind of mainstream medicine and mainstream science that this formalism around double-blind and large N has taken root. And in this age of personalized medicine that we're entering into, where everybody is really different and knows what those differences are by understanding their genetics, I think we're going to have to move back towards this older form of knowledge acquisition, just because the personalized treatments won't be able to find your cohort for the large randomized blinded trials that we've been able to get in this current era where drug treatments are seen to be applicable for everybody in the same way. And so it's something that I think that this quantified self movement is on the right track on, and trying to revitalize this understanding of anecdotal data and personalized data. Seth Roberts' Better Mind experiment, though, is one that has a sad ending and talks about some of the risks of this stuff. So he was convinced that lipids were the key to having a good, agile mind, and had taken this self-driven experiment to do some math problems every morning in this standardized body where we throw them different problems but of the same type and match for difficulty and stuff, and did this very nicely controlled study where for a period of time he did not take any lipids with breakfast. Then he took coconut oil, and then he took grass-fed butter, and would alternate these to match practice effects, and showed some graphs, and this was at the end of, I think, a year and a half of, almost 2 years of a study that he presented at this quantified self meeting. Became convinced that on the mornings when he would have half a stick of butter with breakfast, that he was able to do the math problems much better and was much sharper and feeling much better. So he was at the point of wanting to recruit other people to participate in this with him. And a cardiologist in the audience stood up and went, "No, don't do this." Sadly, about a year after that, Seth died of a heart attack. And they, of course, don't know that it was due to his Better Mind experiment, or that had any role to play. He had some congenital heart issues. But it is a good touch point story to talk about, though, that as you think about these biohacking endeavors, it's collecting information, but it's collecting information potentially at a personal risk. And the responsibility of people working in the biohacking space is to make sure that you're the one understanding the risk. You can no longer foist that responsibility off on the paternalistic medical system to protect you from yourself. That you can do anything, it's your right to do anything with your body, but you're responsible for what you do with your body. And so that's kind of the core of a lot of this, and why I so strongly defend people's rights to do this, is that if they accept the responsibility, they also accept the rights. You don't have to go as involved as injecting things in your eyeballs, or eating a stick of butter every morning, or implanting things under your skin. And in fact, actually, the other neat point I forgot to mention about the implanting things under your skin is Zoltan, the guy whose picture that is actually a candidate for a U.S. president in this coming election. He's the transhumanist candidate. So it's kind of cool. We've gone mainstream enough that our presidential candidates in the U.S. are injecting things under their skin. You don't have to go as extreme as injections and things like that, though. North Paw is a neat project where these folks, I believe they're actually out of the Toronto Makerspace originally, although one of them has down in the San Francisco Bay Area now. But he built this belt that no matter where he was, once a minute it would buzz gently on the north side. It had a compass in there and a bunch of buzzers around it. And so wherever he was, it would buzz on the north once a minute. And it was to try and train his inner sense of direction. And I found that it really worked, that after wearing this thing for 6 or 8 months, he always knew where north was, even without the belt on, that he'd had this very ingrained sense of direction that he never had before. And I find it a really neat example for a couple of reasons. First of all, because, of course, how it was built is with an Arduino and some buzzers and a little compass module. So it's technology that, I mean, the multiples are sitting back in there in the electronic space just ready to be put together in this project. But that his end result was out of putting together these fairly simple components and wearing it in a very risk-free fashion. He has a sense of direction now that he didn't previously have. And so you have an example of people training really kind of basic capabilities with very simple hardware enhancements, and doing so in a way that really has no risk associated with it. I do a lot of work with electroencephalography, EEGs, and the OpenBCI example here on the upper right-hand side is one that I'm really inspired by. These folks decided they weren't happy with any of the EEGs on the market, and went and designed their own and did a Kickstarter campaign to launch it, and now have a very effective 16-channel EEG for under $1,000 that has an entirely open source stack. I'm not going to go into too much depth on electroencephalography in here. I risk getting drawn into the very well-traveled threads of my PhD thesis, and you might see me go into to little shocks of horror and stuff. It's really close to the end on this thing. I'm really sick of that topic. But it is a neat area. And it's neat both from this risk-free training perspective that through EEG neurofeedback, you can really do some neat things. For instance, this Muse headset is a little $300 headset out of a company called Interaxon in Toronto. And it pairs with my cell phone. I turn it on and wear it on my forehead like that. And has a little biofeedback routine that it goes through, measures alpha wave activity, and can tell how deep of a meditation state I'm getting into. And particularly as I get stressed and anxiety ridden about this never-ending PhD, having that 20 minutes a day to go into that meditation state and actually have some direct feedback that I'm hitting an appropriate meditation state, that I really am relaxing. And even though I think I'm spinning my thoughts and worrying about all the things that have to happen in the day, I've got a very direct measure of how effective I've been in letting go of that stuff and relaxing. Lets me enter these states that are kind of mini middle of the day vacation states. And so it really is quite effective. What I'm doing with the EEGs as part of my thesis research is not the slow wave meditation related responses, but rather direct responses to stimuli. So we have participants spell words on the screen based on their brain waves, and they have to concentrate very hard in order to do that. And the sustained concentration is therapeutic for people with attentional conditions. There's another route to go with electrophysiology, though, and that's on the input. EEG is kind of the output side of electrophysiology, and is very safe in the sense that you're not actually putting things into the brain. There are some risks. I mean, if you do a biofeedback protocol that is rewarding being anxious, or rewarding thought patterns you don't want to reward, you can get locked into those loops, and you can make unfortunate psychological impacts on yourself, but you can always get out of them. So it's not totally risk-free, but it's largely risk-free. Transcranial direct stimulation is the process of using small direct current through brain regions. And what it's doing actually is reducing the activation potential of those regions that the current is passing through. Now, in some cases, this can be really good. There's again, U.S. military, they're deep into a lot of this stuff. There's a really good podcast series where one of the people on the podcast went to sniper training at one of these military bases, and sat down, and when she first sat down, and I thought, this is impossibly hard. I can't kill all these targets in this virtual reality environment. And then they hooked up the TCDCS and TDCS, I think I had an extra syllable in there, and found that she thought they had turned the difficulty down and everything was slower. And no, in fact, she'd just been stimulated in the areas of the brain that they were trying to stimulate to make her a better sniper. So this is kind of cool and kind of scary. The part that adds the additional kind of maybe thing to it, though, is that after she left for about the next 2 days, she felt like she was on withdrawal from a drug, that her capabilities were enticing enough that she wanted to be back on hooked up to the machine. And the fact that to build these, all you need is a 9-volt battery, a little bit of circuitry, and a couple of saline-soaked pads to stick to your skull. It's the age of electronic drugs is here if you want it. They found, though, in a lot of later studies on this TDCS stuff, though, that the location that you're stimulating is critical, and that we just don't know enough about where we're stimulating yet. And so it has gone into some of the bigger blinded trials on this stuff. And the initial optimism of the people pushing this technology has not fully borne out. It has in some cases. If you do it right, it can have some effects, but the world's more complex than initial optimism can lead you to believe. And that's one of the other dangers that comes out of these N of 1 and experiential studies, is that it's really easy to fool yourself. And so that's kind of where I'll leave the biohacking thread of the physical cybernetics biohacking thread. is that great power, it's entirely within your right to do all this stuff. Be careful of your responsibilities, be careful of fooling yourself, be careful what you're claiming for things. But I play with a lot of devices that go down this path. This is a Bluetooth blood pressure cuff, for instance, that I find really useful. I was having the doctors telling me I was having high blood pressure, and in fact, using that for a few weeks, I'm now pretty convinced that I was having a white coat anxiety. It's only high in the doctor's office. Everywhere else is just fine. So there's a lot of things you can do with all these new devices, and just do it responsibly. Let's move from there into kind of the core of what I'm finding exciting about biohacking these days. That is genetic engineering. And the intro to this piece of the discussion is that science undergoes this progression where initially it's really, really hard. You've got to really work to get every little piece of the picture. And it's in biology, you can see this, that, turn of this last century, Gregor Mendel with his peas, this monk spent years counting peas and the variations in those peas. And did he get anything useful and applicable out of it? Well, for scientific learning, yes, but maybe for pea breeding, yes. But it was on these long, painful, drawn-out timescales. As sciences mature, though, they become technologies. And as they become technologies, they become infinitely more operable. What I mean by that is, let's look at the progression of physics and the scientific understanding of electricity. And how that's gone through a progression of turning into electronics, which is plainly at this point a pure technology. And you can go and take your Arduinos and take your chips and your resistors, plug them together without having really any thought about what's happening at the electron level. What are the physics properties of what's going on here? Well, no, you're plugging components together, and you've understood this on a higher, more abstract level. As you climb this abstraction tree with a science, it becomes a technology that's more directly operable and can be exposed to many, many more people. And biology is hitting that point right now. It's not nearly as far down that path as is electronics. You really can't do synthetic biology at this point or genetic engineering at this point without thinking about the underlying scientific principles. So it's not like electronics where you don't have to have any conception of the underlying physics. It's nice to have it. I still like to have that conception, but it's not absolutely essential. With biology, it's still essential to have some of that conception, and it's just because it's not as mature. But it is getting mature. So iGEM that I mentioned here, and yeast or bacteria are the place to start. I used to have a glowing cat picture on this slide, and it was causing a lot of people to be confused and think that in the biohacker spaces we're actually making glowing cats and working on eukaryotes and doing human genetic engineering. And, maybe someday, but it's not there. And there's a whole lot of ethical levels and a whole lot of legal levels and a whole lot of discussions to have before you go there. But in terms of yeast, in terms of bacteria, it's totally fair game. The bacteria species we use is E. coli, which you might have negative associations with through people getting sick from their hamburgers. In actuality, the E. coli that we use in the lab is a lab attenuated strain. And I had a prof in one of my early lab courses actually take a swig from a beaker of this stuff, and it was really gross, and everybody went, ooh, and remembered that as, okay, it really can't hurt you, but ooh. So the goal is, and I'll talk about this in a little bit later slide, is that in a hackerspace, we do nothing that is not biosafety level 1 or safety category 1. This means it poses no risk to human health or environmental release. Within those constraints, though, we can do an incredible number of neat things. iGEM is, iGEM is a competition that MIT sponsors every year, and it's now open to hackerspaces and members of the general community and not just university students. 2 years ago was the first year that they did that. And it stands for International Genetically Engineered Machines. When you join the competition, they send you 384 well plates. Each of those wells has in it a different genetic block. Can treat them kind of like Lego blocks or like electronic components. And so a block might have a sensor. It might be able to detect melamine. It might be able to detect arsenic. A block might be a protein production site. It might produce a red fluorescent protein, or it might produce a a spectrum of chromatic colors if it's a more complex circuit. A block might be a ribosome binding site. This is something that when you actually produce the protein, when the DNA is on its way to being turned into a protein, that ribosome binding site is where the machinery hooks onto to actually build the protein. And you can build these ribosome binding sites so that they're temperature sensitive. So for instance, one of the projects we did in the first year that we did a Victoria entry into iGEM is we took these blocks together and built a bacterial thermometer, where if you grew it up at room temperature, it would produce a green fluorescent protein. If you grew it up at 42 degrees or over, this temperature-sensitive ribosome binding site would break its little hairpin loop, so it would begin producing lots and lots of red fluorescent protein. So grow it up room temperature, it glows green. Grow it up in hot, warmer conditions, it glows red. It only does it once. It's not like a button you can shoot with your laser and turn red or green like we would have electronics. But biology isn't near as a mature technology as electronics. And we're moving that direction where we can do all of these things in biological formats that we just couldn't do in electrical formats. Sometimes you have different tools that can achieve the same end. Things really have progressed since then in iGEM, and the winning teams in the competition are doing projects that just a few years ago were these multi-year, multi-million-dollar research projects that as soon as you start componentizing the factors that go into it and start being able to move these different genetic components around, to rearrange them into the circuits that you're designing on the computer, and then executing and dropping into a bacteria, everything moves faster, right? And so the pace of genetic research is really expanding with this componentized approach to synthetic biology. I'll talk a little bit about what's going on at the actual underlying level here, but this slide's mostly for us to come back to if people have questions and want to drill into to this bit in more detail. Being a bit conscious of time, I'm going to skip over pretty quickly, though. The ampicillin resistance gene that you see over here on the red is the thing that is actually allowing us to do a lot of this manipulation. We use ampicillin, kanamycin, and chloramphenicol as the three antibiotics that are used in the iGEM assembly methods to ensure that only those bacteria survive that have taken up the new constructs that we're putting into the bacteria. And so we'll alternate the bacteria's antibiotic resistance with each new part of the construct, each new block on the chain that we put into the bacteria. We'll change the antibiotic resistance to make sure that anything that hasn't picked up that new piece gets killed off. And by progressively controlling the conditions under which the bacteria will live or die, you end up setting up this evolutionary process where only the things that have assembled in the way that you want to assemble will survive out of that. I gave this talk at a TEDx Victoria a few years ago, or this part of the talk a few years ago, and had someone come up horrified after the talk going, but is this ethical? Think of the bacteria. And bacteria evolution and death is very different than you or I. When you drink a cup of coffee, you're killing tens of thousands of bacteria. And so yes, I have actually thought of bacteria, and I don't care. Some legalities around this that Bear mentioned. And I talk about this in part because I really want to encourage you guys to play with some of this stuff in the hackerspaces. There are some kits out there that make it really easy to get started with some of this, or come down to Victoria and take our Build Your First GMO class that we do periodically, usually about once a month until I get my thesis finished. I'm not teaching it again, but by February, I will teach it again. But in the Create Your First GMO class, we take a green fluorescent protein from jellyfish and insert it into bacteria and talk about cell transformation and all the parts that go into to that. Really approachable. We've had kids as young as 5 come in and create their first genetically modified organism. We use it as a good way of talking about genetically modified organisms and why they're not necessarily all bad like are portrayed in the media. If they are driven by corporate concerns and profit-making concerns, there's a lot of bad things you can slip in there. I don't know that I'm a huge fan of GMO foods because a lot of that has been how do you make it more profitable? How do you make it more industrially accessible? But if you're putting in changes that you want to put in and using genetic engineering for things that are really coming from grassroots concerns, people wanting to change the world in the ways that are appropriate and beneficial to them, GMO is not a bad, scary thing. So Create Your First GMO, we create this jellyfish glowing bacteria. Totally safe. This is a safety category 1 activity. What I mean by this is an activity that poses no risk to environmental release or human health. And to date in Canada, this is entirely unregulated. In Europe, this is not true. In Europe, you actually have to get a transformation license before, even in these lab attenuated strains of E. coli that can't survive outside the lab, before you make any genetic modifications in one of these. I see this as a serious competitive barrier to European science and European science education. That if you have these activities that really are essentially zero risk activities and you're legislating against them, you're stopping people from even taking that first step of satisfying a scientific interest. And so I really see our unregulated use of biosafety level 1, safety category 1 activities in North America as being a serious advantage to the system. That having been said, the Canadian Health Authority is introducing some guidelines for safety category 1 organisms. They're going to be optional guidelines. There's no force of law behind adhering to these guidelines yet, and I'm all in favor of optional guidelines because you do want to do things to stay safe and inculcate good lab practices even in safety category 1. It's your training ground. You don't want people to get sloppy there if they're going to go on to things that could hurt them later. And so as long as we keep this in the category of these are guidelines to stay safe and good practices, wonderful. If we cross that you have to go get licensed to do these risk-free citizen science activities, that's where it starts to get iffier. And it doesn't look at this point like we're crossing that license path. If you do want to go up into safety category 2 activities, you do have to get licensed at this point. And again, I think that's fairly appropriate. And the safety category 2 activities are things like Staph aureus, which all of us have, but some of us have multiple antibiotic resistant Staph aureus. In one of my microbiology labs at UVic, we did a nose swab on everybody in the class. There were about 40 of us in that lab, and out of those 40, 6 had multiple antibiotic resistant Staph aureus living in their nose all the time. So even just swabbing your nose or swabbing your cheek and culturing something up in a dish is a safety category 2 activity, and rightly so, because you don't necessarily know what it is that you're growing up. Whenever you don't know that what it is you're growing up is absolutely safe for you and the environment, you're in safety category 2. I would argue that at the moment it is not actually correct to have open biohacker spaces involved in safety category 2 activities. I have a safety category 2 lab at Makerspace, but it's actually a space that I rent separately from Makerspace, have a locked door on it, and I control access to that space. And unless there's somebody that is actually responsible for controlling access, controlling the activities in a space, legally it's incorrect to do safety category activity to activities, and I think morally and ethically it's incorrect as well. And so if you do start doing biohacking here at Nanaimo Makerspace or other hacker spaces, one of the things to really keep in mind is keep the safety profile foremost in your mind. Make sure you're doing activities that are appropriate to do in uncontrolled spaces. Sorry, that's enough legalism. Let's talk about genetics and health some. So I'm kind of leaving the thread of synthetic biology in the lab to more personal genetics and things that at this point are not directly operable. I'm not making changes to my own genetics, but here in a couple of slides, we'll talk about changes to your own genetics and how this day is coming very, very quickly, for good or ill. But some of the things that you can learn from your genetics and make them personally operable. This is biohacking just like anything else. If you talk about shortcuts to human health and performance, well, one of those shortcuts might be wearing a belt that buzzes to teach me something new. One of those shortcuts might be using knowledge of my genetics to take a supplement that works great for me, but might not be necessary for the rest of you in this room. So I think it's a form of biohacking that's really just applying knowledge about biology to the way that you live your life. 23andMe is a company in the U.S. That's been in the news a fair amount lately for their battle with the FDA, which was induced by pure stupidity. They do a SNP test, single nucleotide polymorphisms, single base changes in DNA. Now, this single base might not be important in some cases, but DNA moves in blocks, essentially. So if you have this single base change, it's likely that that's a marker for other DNA changes around that have more than a single base impact. In some cases, a single base can have an impact too, of course, but it's really representative of larger changes. And so they, when I took the test the first time in 2009, I believe it was, they were giving a report with about 500 or so different health claims for what these SNPs meant. And they got away with that until early last year when the FDA said, wait, you're making health claims. This is not allowed. You don't have the evidence to back this up. And so now they're going through the process of trying to convince the FDA that they do have the evidence, that they're setting up these tests properly. But in the US, you can't actually get access to all of that health information. You can still do the test. They give you the raw data, but they can't tell you the health impacts of it. They're focusing on genealogy and ancestry and things like that. In Canada, we can still get access to the full test. But there's actually a service called Promethease that is a much more in-depth analysis of the health aspects of this data that is not encumbered by the FDA regulations, and they're not a for-profit enterprise. It's essentially crowdsourced genetics knowledge. And so I'm going to skip back and forth over this a little bit, but in 2010, I gave my sister Alzheimer's for Christmas. This was not a happy Christmas, by the way, or the months following that Christmas. There's a particular gene called ApoE4 that is the most significant risk factor for Alzheimer's. I'm a ApoE4 heterozygote, meaning I have one copy of ApoE4. This gives me about a twice lifetime risk of Alzheimer's over what most of you in this room have. The background risk is about 7%. It's about 14% in me. Due largely to a different lipid metabolism. I process fats differently than people who do not have one of these ApoE4 genes. My sister, unfortunately, turned up a homozygote for ApoE4, which is really bad. This is right now giving one about a 70% chance lifetime of Alzheimer's. So it was kind of grim thinking about this, and they weren't necessarily happy that I had given these 23andMe kits out as Christmas presents. My dad actually never did end up taking the test. He didn't want to know the results. At this point, though, a few years down the road, looking back, my sister's made some lifestyle changes. She's tracking with her doctor what some of the other risk factors are. There's been some research out showing that actually about 90% of the risk factors are modifiable, that they're lifestyle modifiable. So genetics is not determinism. It does not guarantee that you're going to get some condition. But it can certainly predispose. So I don't really think that was the Christmas I gave my sister Alzheimer's, but boy, it was uncomfortable for a little while there.
Are those results 100%?
They're not. This is another thing that you have to think about, is that the accuracy of the results is pretty good. I think it's about 99.6% on an individual base region that it's going to be correct out of 23andMe right now. But their current chip samples, I believe it's 700,000 different sites. So that 99.6 turns into, there are going to be a couple dozen that are wrong in each individual. And are they going to be on ones that determine hair color? Oh, I know it's red. Or are they going to be on ones that show Alzheimer's risk? Kind of luck of the draw. It's getting better, but it is something to keep in mind. And then the other one, of course, is that it's not determinism, that lifestyle factors impact a lot of this. Let me actually skip out of the presentation at this point to show my Promethease report and talk a little bit more about that. So here is and actually let me show the front page first. So on the front page of the Promethease report, there are a bunch of different things, good, bad, interesting results. On the good side, I have a lower risk than usual of atrial fibrillation, a reduced risk of late onset, adult onset, or asthma rather, a lower heart attack risk in some ways and bigger in others. So they're real kind of hard to interpret. You got to jump around and balance all the different factors. This is one of the hard things about genetics at this point, is that it's very, very interrelated. On the bad side, though, I have a 7 times higher risk than most of you going bald. This might be true. I'm starting to do it. They're probably right on that one. Here's a factor that gives me a 1.9 times increased risk for coronary artery disease. I think it was a 66% chance of normal on the other side. And so these all weigh off, and again, interpretation is a huge problem on a bunch of these. Problem metabolizing NSAIDs. So things like ibuprofen. I've got to be careful about not taking too much ibuprofen or the other NSAIDs for pain because they don't break down as well in me. This is actually one of the things that I think is really valuable about this report is if we look at medicines, there are a bunch of things in here. Antidepressants. I react very, very badly to some antidepressants. For instance, paroxetine or Paxil. I react just about as badly as you can react to it. I would not want to take Paxil if I needed to take an antidepressant right now. Effexor is another one that is in that list. Venlafaxine that I would not choose as a first choice antidepressant to try. That otherwise I might have been kind of blindly guided by the doctor that I went to. And why don't you try this one first? I've had good luck with it. Well, yeah, for most people maybe, but no, not for me. Just having this knowledge of your genetics is in fact a source of power in negotiating with the medical establishment and what the first course of treatment on things might be. And it's not the only thing. It's there are different factors in there. Interpretation is always a problem. But if you have no data to start with, this is at least a data point to start with. So one that I found personally actionable on this, and I'm going to first jump over to this other service that was going to talk about first, but kind of missed. But Heart Health, Erie Vale. This is a program that is run out of Seattle. Leroy Hood was one of the founders of the Human Genome Project, where they first sequenced the human genome. And started an institute at the University of Washington called the Institute for Systems Biology, where they were trying to track all of the different genetic impacts of health and kind of what you could do about them. It built May or so of last year of 2014 rather. He left University of Washington to start this company called Erie Vale, primarily because he wanted to do a commercial application of this information. So what happens now with Erie Vale is you pay them $2,000 until the end of the year. It goes up to $4,000 in January to essentially participate in their clinical trial. And this is why it couldn't happen at the University of Washington, is that the ethics of paying to be in a clinical trial are sometimes maybe suspect. I decided to participate in the program because for that $2,000, one of the things you get is a complete genome sequence, which up until earlier this year was a few $1,000s on its own, as well as a whole bunch of blood test results and a monthly phone conversation and weekly email conversation with a counselor, trying to make diet and lifestyle changes in response to the blood conditions they find and the genetics that you show to achieve optimal health and wellness. And one of the things that came out of this program for me was that I have an elevated homocysteine level. Homocysteine is an independent factor for heart disease. It's an inflammation marker. So this is something that I actually could have found out on my own from the 23andMe information. I didn't know to look for it. It's one of those things that's buried down in the morass of things I didn't think to notice. But on my Promethease report, it shows that MTHFR, which is the enzyme that breaks down homocysteine, is only 60% efficient in me. So I'm not breaking down homocysteine nearly as efficiently as most of you are. Now, this is one of those conditions, though, that not being something that genetics is determinism, and this is just something you got to live with, there's a really easy solution to this. B-complex vitamins. Or I could drink more beer, I guess. But B-complex vitamins are the building block for this particular enzyme that breaks down homocysteine. And so there's very good experimental evidence that if you take additional B vitamins, it boosts the functionality of these enzymes up. And so when we take the next blood test in the series at Erie Vale in February, I hope to have seen those homocysteine levels completely normalized. Just little examples of finding specific places in the genetics where they interact with your lifestyle, supplements, exercise, whatever else, where you can make really big changes that are pushed by a very little piece of genetic information. Let's pop back to slides. Oh yeah, so there are a couple other places that you can get a genome done. And when I say a genome sequence, this is different than what 23andMe is doing. 23andMe is looking at 700,000 SNPs, single nucleotide polymorphisms, and trying to infer from those SNPs what's happening with your genome. A complete genome sequence is sequencing the 3.2 gigabases of your genome, and so you have every letter of your genetic code. Is almost right. In reality, your genetic code is changing in all of your tissues all the time, and so you don't have one genome. You have a genome that's specific to a tissue, and that genome sequence doesn't give you the epigenetic information, what genes are turned on and off at any point in time. The picture is always more complex than we make it. But at the surface level, a genome sequence taking some core part of your tissues, in this case a cheek swab, so they get skin tissues, and sequencing that so they across the different samples, they do an 80 times depth sequence. They get a consensus across those 80 different sequences. They do essentially and say, send me a hard drive with my 3.2 gigabases of DNA on it. Then I can actually search anywhere and know what is in there, rather than just being restricted to the SNPs that 23andMe ran. And that's one of the things I actually find useful about this information, even from 23andMe, is that when a new study comes out, I dig into the raw data and go, oh, RS13574, do I have that? Oh, it's not on the chip. Rats. I don't know how to apply that study to me. Maybe I'm just selfish. I want to apply all the studies to me.
No good to know too much.
That's right. That's right. The other thing we should talk about with genetics is the genetics in your DNA is actually only about 1% of the DNA information in your body. 99% of the DNA in your body is not yours. It's the organisms that live with you, your gut bacteria, your skin bacteria. You might have a fungal infection. You might have a helminthic worm living inside you somewhere. All of the other creatures that live with you actually contribute 99% of your DNA. And this is kind of amazing when you think about it, in the sense that when you eat something, it's going to your gut with the largest collection of bacteria there. If they weren't there, you couldn't digest most of what you've eaten. Your bacteria are actually playing a commensal role, a cooperative role with you. They're breaking down what you eat and making it available for you. And in return, you're providing a nice, safe, warm place for them until you drink that cup of coffee. But the collection of bacteria that are down there have a very integral role to your health. Companies like uBiome or Second Genome or the American Gut Project at the University of Colorado Boulder are all looking at this colony of bacteria that you carry with you and what they can do for you. And so jumping back over to the American Gut first, just because I like the way that it presents things. So here is my gut from the American Gut Project. Very non-diverse, but then Michael Pollan is also non-diverse. It's funny, they compare me to Michael Pollan, and that's their sample of an individual. Very much less diverse than the average, though, and they can make some claims about some of the populations of Firmicutes versus Bacteroidetes. They have an influence on obesity, for instance. I'm carrying a few extra pounds. If I could change my gut bacteria, this might not happen. But they talk about some of the averages in the population and can compare myself to it. Well, that's kind of neat, but what do I do with that information? Some of the other places, like uBiome, get a little bit more operable on that. And I can also track over time. I've been doing uBiome studies for a while now. After late June of this year, I actually had a couple episodes where I had to be on antibiotics for a bit. You can see that my gut bacteria have plummeted in their diversity. That a lot of these species here that were there before the antibiotics just aren't there now. And getting them back after a round of antibiotics is really tough. Taking oral probiotics is just really not that effective at restoring gut microbial populations. They've got to go through a very acidic stomach. The populations that are there really entrench and make it difficult for new species to populate. Things like fecal microbiota transplants are a lot more effective, but and it's not really serious enough for that yet, thanks.
How did they get this data from you?
They got this data from me in a swab off my tissue paper after I used the toilet in the morning. The second genome that I don't have the data back from actually had a little bit more involved process that required a little plastic shovel, but we won't go there. So in different ways. But yes, they do sample the poop. But there's a big difference to my eye between sampling the poop and blendering somebody else's and putting So it's really effective though for a lot of conditions. Even though it's kind of gross to think about, they have achieved over a 2/3 remission rate for C. difficile infections by fecal microbiota transplants. Some studies have shown up to an 80% remission rate in Crohn's disease from fecal microbiota transplants. These very, very serious conditions are treatable by changing gut microbiota. And so squeamishness really, if I were in one of those situations of having a serious disease that needed that treatment, would not enter into the equation. If I had Crohn's and the choice was between getting part of my intestine surgically removed and having a colostomy bag or shooting someone's poop up my butt, I know where the poop would be going. So let's pop back to leave poop for a minute. And we talked about Aryvale, talked about Promethease. So let's move into modifying genomes. We've got all this stuff, and I'm a little bit conscious of time here, so I'm going to skip pretty quickly past some of these last pieces and leave it for you guys to ask on questions. We've got all this information about genetics, and what's it do for us? Well, I can make lifestyle changes. I can change my supplements based on that knowledge, but it's not until I can actually modify my genetics that I'm really happy. Not until I can get in the machine and take the parts I don't like and snip them out and put in something that I do like. We used to think this is pretty much impossible. The ways that we're doing it in the lab with bacteria require that you have all these antibiotic resistances, and you have to set up the right evolutionary conditions. Enter CRISPR-Cas9. This is actually a bacterial defense mechanism that was discovered just a few years ago. Well, actually, it was discovered about 20 years ago, but its relevance to genetic engineering was just discovered a few years ago. And it used to be that the bacterial defense mechanisms were all thought to be unchangeable enzymes. So for instance, EcoR1 is an enzyme that E. coli produces that snips DNA that most other bacteria have, but E. coli happens not to have. And so it's an offensive mechanism to make sure that when you have multiple bacteria together with E. coli, and E. coli is releasing this enzyme that snips their DNA but not its own, that it has a competitive advantage, so more E. coli will live than the ones with the snipped DNA. The thing that's neat about this CRISPR-Cas9 mechanism is that it's flexible. That what is causing the location of the DNA snip to occur is not inbuilt into the enzyme, but instead fed to it by a piece of RNA. It's essentially the enzymatic version of a programmable computer. Think about that for a minute. It's the enzymatic version of a programmable computer. That you can retarget this enzyme by feeding it a new piece of RNA or a new piece of data. Well, that's what we do with computer programs all the time. It's like going from a mechanical adding machine that just adds to a von Neumann machine that we handed its program in a flexible, changeable way. And we can do a lot more with our computers than we could do with those mechanical adding machines. The same is true with a genetic modification. That when what we have to do is implant the machinery to read these codes stably, and then feed the codes to make the edits, the whole world changed as to what's capable in here. Now, it's still hard. In order to do that in URI, you've got to think of a vector to get these changes in there. It's probably a viral vector, so people have been really successful at doing things like taking an adenovirus that gives you a mild cold kind of symptoms, putting a payload inside that, putting a targeting mechanism to go target only a specific piece of tissue, and then making an animal catch that cold. But it's a really messy process, and to think about trying to infect every cell of every one of our bodies, it's just kind of an impossibility. Fortunately, our cells turn over a lot, and so this is where stem cells come in. And if, for instance, I had a chronic kidney condition, I've depleted my stem cells in my kidneys, you realize genetically, oh, here's the factor that has made it become so chronically depleted there. Can we take your stem cells out, modify them, snip that piece out, grow back up your stem cell population, re-inject that stem cell population? Well, here there's going to be a selective pressure for the modified stem cells to continue to do their thing. And so you've got an area where you can think of a process for making targeted genetic changes, re-implanting those changes, and having evolution do the job for us. Again, it's not changing all of the cells in our body, but it is a way of thinking of making these targeted genetic modifications. You can also think of this as being very useful in terms of organ printing. I'm not even going to go into organ printing in this talk, not enough time for it. But there are ways to go back and treat the human somatic genome, the genome that is not the reproductive genome. Reading and writing genes is getting really, really cheap. Fly over this one. This is Moore's Law, the white line at the top. The cost per genome is the green line there. It seemed to parallel Moore's Law for a few years there between 2011 and 2014. I was getting nervous talking about this exponential increase. And then we hit third generation sequencing and the advent of the HiSeq 10X sequencer from Illumina, and we've dropped off a cliff again. We're way, way faster than Moore's Law. Here is the neat possibility you should think about, that writing DNA has not been following this type of increase. It's about 15 years behind reading DNA. But we are now starting to see companies like Twist who are doing writing DNA on a silicon platform, who are doing so at the picoliter scale, in the nanoliter scale, not at the milliliter scale that synthesis has been happening at today. And it's dropping off a cliff in terms of pricing now as well. And it's kind of neat to think about market opportunities, to think about a systemic change when you know you're at the start of a 10 or 15 year exponential process. We know that over the next 10 or 15 years, it's going to get exponentially cheaper and easier to write DNA. What do you do with that? Well, you're not stuck in the lab doing these one at a time componentized building block changes anymore. If you can push a button and have half a genome pop out and drop it in. We're not there yet. It's still $64 million, even at the 2-year, 2 cents a base increase Twist is proposing. And we're not going to push a button and spend $64 million on printing a new piece of DNA when we can cut it together with enzymes, but we're heading that direction. Think about exponential changes. Think about knowledge changes, that with this becoming information science, and biology really is an information science at this point, what can we know, what can we do as we know more? There's a supercentenarian project looking at people who have lived over 110 years. What's different about their DNA? What's different about their lifestyle? Can you put the two together? This is giving us information on what the super long lived among us are doing differently and how they're built differently. There's a Methuselah Mouse Prize for making the most longest lived mouse. Caloric restriction is always known to be useful for promoting longevity, but it kind of sucks to miss that piece of birthday cake. What can we do to medically and pharmaceutically promote the same type of results? Metformin, there's actually been a recent study that showed that people on metformin with type 2 diabetes that you might expect to live shorter lives as a result are actually living longer lives than their non-diabetic cohorts. Possibly the influence of metformin in stimulating some of these same pathways that are involved in caloric restriction. There's a dog study using rapamycin, which has some nasty side effects. It's an immune dysregulator. It makes you prone to conditions that are very non-ideal in terms of your immunity. But put a dog on rapamycin and it lives 25% longer than it otherwise would. So longevity is malleable, and we're going to continue to find better and better ways to play with longevity to allow all of us and all of our children to live longer. Here's the one, though, that is really making my mind blow on this stuff. Beijing Genomics Institute has about 25% of the world's sequencing capacity. They have whole cities at this point devoted to biomedical research. They founded a new city 3 years ago that has 2 research universities, 2 major hospitals. They started the city from scratch just to promote biomedical research. We just don't pursue biomedical projects at this scale in North America. And we're going to be in some serious hurt as time goes on at not pursuing it at this scale. BGI has a cognitive genomics program that is a 5-year program that at this point intends to sequence about 10,000 people to determine the genetic correlates of intelligence. the head of this program came down to give a talk at Singularity University in the Bay Area that I attended just this last summer, and they're into their second year and accelerating into it. And some of their early data on this is both amazing and terrifying. Amazing in the sense that they think that they've identified the start of a 100-gene network that they think of as sand in the machine, basically these little deleterious mutations that all of us have. And if you think of the evolutionary probability of 100 genes all pointing in the most advantageous direction, well, it's a factorial. So 100 times 99 times 98 times 97, all the way down to the bottom. The chance of any of us having all 100 of these genes pointing in the same direction is just infinitesimal, even over evolutionary timescales. But if you know what the right direction is, and you know how to make those modifications at an embryonic stage to ensure that somebody has all those pointing the right way, they're positing that the effect of this 100-gene network is about a 15 standard deviation enhancement in IQ. This is an IQ of well over 300 IQ points, since the standard deviation is 15 IQ points in humans, centered around 100 right now. Think about this, that if a population here in a generation or so, and this is a 5-year study, a population here in a generation or so is 3 times as smart as the people being born here. We currently have a moratorium in North America on the use of genetic engineering for human germline enhancement. There is no such moratorium in China. There's a global competitiveness question that will very, very quickly arise here. And in some sense, our hand is forced by just the rapid advance of technology here to keep up or become obsolete. Designer babies. I talked to those points right there. So what's the role of Hackerspace in this? I think there are a number of roles. The first is education. Things are moving too quick. You can't stop learning when you leave school anymore. That's what everybody's here for, is to learn more technologies, more neat things that you can apply. Pushing the envelope. You don't have these discussions in most other venues about what can you do with technology and why is it a good thing for us to do it, and why is it a good thing to push the envelope and push society on technology. If this is purely a corporate function, it'll be driven by what can make money, and that's not what's probably at the core of what most of we want. We want to make better lives for ourselves, for others, for the environment. It's not purely a corporate-driven question. Reskilling, that biology as an information science makes a lot of the computer expertise that a lot of us in this room have applicable to a new domain where it's very, very badly needed. But you have to know something about the biology underlying that before you can apply it to that domain. Along with reskilling, think about those exponentials. If you knew in the 1970s, the 1980s, that Moore's Law was going to carry on for 40 more years, there's a whole lot of things you could have done to position yourselves to be at the right point on those curves to make money, control industries, make a long-lasting change in the world. We know right now that we're at the start of an exponential period in biology. It's a good train to hop on. And the grassroots of just what would you like to see engineered and how would you like to see it? I bet it's not the antibiotic or the pesticide resistances that the big agribusinesses are putting genetic engineering towards today. And we'll leave it there. Nobody is asking any questions, so let's have people hit me with questions. Yeah, Joe?
It's a lot. Where can you find those My First GMO kits, and about how much do they run?
Yeah, so come on down to Victoria, but otherwise North Carolina Biological is one good supply house that will send to individuals. The materials to make such a kit are actually not terribly expensive or hard to get, but there's a real problem in getting individual residential addresses to be delivered to by the scientific supply companies. This is one place that hackerspaces are really useful in that there's a business address at the hackerspace. And so at Victoria, we've established ordering relationships from folks like Sigma, folks like Cedar Lane, the different biological supply houses. But the easiest first part, if you want to get the instructional material along with it, is Carolina Biological. Yeah.
So I followed 23andMe for a while. I think when I first looked at it, it was $1,000. I just looked it up now, and it's down to $200.
Yep.
Do you expect to continue to see this to dropping, like you've shown some of those graphs?
So we're here at kind of the low end where the majority of 23andMe's costs are no longer running the SNP chip cost. They're instead the employee costs to deliver information, to create their website, to do the FDA processing, to run their business. And so I don't expect to see a kind of low-end consumer-level testing to be on an exponential growth curve. At the higher end, doing a full genome, we're still at something more approaching an exponential growth curve on that. We have dropped under $1,000 genome now. The Public Genome Project has negotiated rates with one of the sequencing providers to provide a sub-$1,000 genome to individuals. And so I'll expect to see the full genome prices coming closer to the SNP chip prices. But I suspect that aside from the $100 sale that 23andMe does about once a year and kind of progressive increases, that we won't see it off an exponential cliff anymore.
So when, with this stuff being so cheap to run it, when do you think it'll be that my doctor will send me out, get some blood tests, and while you're at it, I'd like to see your genomes to check a few things, kind of thing? When do you think that'll be common practice? I always figure more information for them, more to, even if, and not only for them to diagnose, but to go, this guy got this, here's his genome, add it to the statistical analysis.
Yeah. So in some conditions, they do it quite as a matter of course at this point. Cancer, for instance, the Genome BC over at UBC Medical Center, any kid who has cancer is getting their full genome done, just as a matter of course, because that's one of their focus areas. An adult with cancer kind of depends on which cancer you have. At this point, if I was diagnosed with cancer and I couldn't get them to run a sequence through the BC Medical Service, I would go to Foundation Medicine, which is a U.S. company that is doing cancer sequencing and making personalized treatment recommendations on that basis, and have been having great success with it. It takes time to get into the medical system, though, and at this point, interpretation of the genome is as much of a cost as is the actual provision of the genome. And medical systems are conservative, and your doctor actually doesn't necessarily want that more information because he doesn't know what to do with it. So this is really where, with personalized medicine, more responsibility is falling on the end user, that if your genome is that much different than your neighbor's genome, it's your responsibility to have that information and be able to go to your doctor and say, no, that's the wrong course of treatment for me, and here's why I think so. Which is really a reversal of the payment structure and the reversal of the incentive structure to what the medical system has had.
Are you the only receiver of the results, or is the report stored somewhere? Is the insurance also involved somehow with 23andMe? I don't know.
Yeah, it's a really good question. So 23andMe is a U.S. company. In the U.S., there is a piece of legislation known as CALEA, which is preventing the use of genetic information for insurance policy decisions. So there's some protection there. 23andMe is keeping the supposedly anonymized information for research purposes. And it's a consideration. It could have a life insurance impact. We don't have that same anti-discrimination legislation in Canada, and so life insurance could look at those results if they had access to them. I think personally that the age of genetic privacy is over, that if we look at these exponential changes in the ease of sequencing, the day when I could have an air sniffer picking up discarded skin cells from everybody who walked through the room and sequence them is not more than a decade away. And assuming we follow the same exponential curves. I've actually put my genome up into the Public Genome Project and made it publicly available as a research instrument. I think that that day of privacy is over, but it's a valid concern. And if you're in a situation where you need to protect yourself from potentially a life insurance company or from legal action in some ways, then it's something to think about. The different companies have different policies on that. If you are paying for a full genome from something like Complete Genomics, you're buying the genome and they don't keep a copy of it. If you're paying the $200 to 23andMe for a SNP kit, they're actually subsidizing the cost of it by using it as a research instrument.
A couple of things. I mean, I didn't watch Scanta recently, but I could have by the presentation. You know, kind of one's dystopia future is another utopia future. So, and physicians I mean, they don't know what to do with the information they have now, and they're well aware of that, and the scientific research that goes along with that. I think we're a long ways away from Western medicine and getting involved at a broad scale.
Right.
For that. Is that your kind of consensus as well?
Absolutely. And there's a lot of patients' rights groups, again, mostly focused out of the U.S. They've got a much more dysfunctional medical system than Canada does. I think we're in a little bit better situation here in Canada just because of size and a more responsive medical system. But in the U.S., if you're under a major HMO, they really actually don't care about you. They care about making the money off your premiums. And consequently, there are much more active patient rights groups. Patients Like Me is a particular organization that, again, is focused around cancer patients. But for almost every medical condition, serious medical condition out there at this point, there's some form of crowdsourced patient community for that condition, with the entire reason for existence being to take some of these scientific advancements and personal medicine advancements and advocating for taking them back to your doctor to get the care that you deserve.
I'd be concerned also as far as Sebastian mentioned about insurance. I mean, the way Canadian insurance policies is you have to disclose everything you know. So if you had it done and you know it, now the ball's now in your court.
Yep.
And so it's up to them to disprove that. But I think that's a real risk, just knowing it yourself. And I mean, just as much as it can be manipulated into adding anxiety and everything else.
Right.
You're taking a risk there, and you need to know that risk before you do that.
You do, and you need to know a couple other things around that risk. I'm getting ready to, when I finish the PhD, leave UVic and was starting to look into extended medical insurance and I discovered that if you are more than 90 days without extended medical insurance, that you are responsible for all pre-existing conditions. So it became very important to me to keep a continuity of extended medical after leaving the university because I now know a whole lot more than I did before I entered the university, and that would be all pre-existing at this point for me.
I had a question about the accuracy of the report. So you've got the 23andMe 700 S&Ps, and then you've got your full genome.
Yep.
Being able to compare the data and see if there's any discrepancies.
Yeah, it's actually 700,000, but I don't yet have my full genome from Maryville, so I haven't made that comparison, but I'm dying to do so. I have done the 23andMe several times now as they've increased their platform, increased the number of SNPs that are on there. The first couple of times I actually did it using pseudonyms. And did that in part because I was worried about having the information out there and hadn't hit some of the same concerns you were worrying about, and decided that I was dead to privacy. But was really kind of neat looking back over that old data because it does a lot of ancestry stuff on 23andMe and told me that I was only 99.4% related to the previous version of me.
I work with special needs adults, and one of the models in autism that was presented over the last couple of years is that the systems model of the brain, where different parts of the brain are all functioning together. Kind of is like a standard normal brain that we all have. But people with autism don't connect different parts of their brain together. They don't operate as a whole. I'm not sure if you're familiar with that.
Yep.
So you mentioned the part about the sniper and the external enhancement or stimulation of certain areas of the brain. What a neat hack is to actually create an external connection system so that now a person with autism can have all the different parts of their brain now available to them in a way that they could function differently than they normally do.
Yeah, it's really fascinating. It's really fascinating. There are a couple of models of autistic brain function at this point, with some of them actually positing overconnection as being as much of an issue as underconnection. And deep brain magnetics, one of the limiting factors of TDCS is that it really only activates surface brain structures. If something's buried under a lot of layers of gray matter there, you're not going to get the surface current to go down and activate it. But you can deactivate very easily and very precisely with magnetics. So this combination of being able to activate electrically or deactivate magnetically brain regions to create this ideal environment for whatever purpose you're trying to use it for is a fascinating area of brain science. They've also done a lot with deep brain structure neurofeedback. Krista Sharms runs a program in the Bay Area where he uses fMRIs for neurofeedback on chronic pain patients. And it's found essentially that a dozen sessions in this neurofeedback fMRI is the equivalent of strong opiates, that they can essentially train themselves not to need opiates for their chronic pain conditions anymore. And so the ability to both mechanically, like with the magnetics, or self-regulated affect our neural function is an explosive area.
Well, some of you might want to chat with Derek on the side afterwards, but in the meantime, I just, on behalf of the group, you know how broke makerspaces are, but we did a little something for you just to show appreciation, because we're really honored that you would take a day to come up here on your own time and speak to us. But more importantly to the rest of you too, if you get a chance, Google Victoria Makerspace and pop in sometime. And I think it's really important, and I think that's what some of us are going to be doing over this next year or so, is really starting to connect all these spaces that are popping up. I think it's really important. And then maybe we'll get live portals. It'd be nice to have a live portal down to Victoria. It would be cool.
Yeah, it would be.
So thank you so much.
Well, great. Thanks so much for inviting me. I'd love to answer questions. I'm going to pee first.
Help yourself to another drink. I don't know if there's